So today the capillary electrophoresis how actually works we will discuss. Capillary electrophoresis is a technique, electrophoretic technique, which is used to separate the charged molecule based on their differential rate of migration when when it is exposed to the applied electric field. So electrophoresis was a technique for separation of charged molecule was developed by Sweden chemist and to separate the serum proteins, and for this work he got a Nobel Prize on 1948. So the migration of charged molecules under the applied electrical field mainly depends on the charge to mass ratio, charge and the size ratio. So based on that, the smaller the molecule and greater the charge will migrate faster than the larger the molecule and lesser the charge. So normally this type of capillary electrophoresis can be used to resolve proteins, since proteins are also charged, vitamins, peptides, amino acids, nucleic acids like RNA and DNA, inorganic ions, and organic bases, organic acids, enzymes, etc. So normally in this particular electrophoresis, the buffer filled in a fused silica capillary tube. So buffer will be filled in a fused silica capillary tube of 10 to 100 micrometer in inner diameter, and a long length of the capillary tube is for 40 to 100 centimeter long, and both the ends of the capillary tubes are immersed in a buffer chamber along with the electrodes, respective electrodes, platinum being used as a electrode here, and higher voltage potential is used starting from 5 to 30 kilowatts of very high voltage is used here. And as a result, the sample also has to be injected under pressure or vacuum into the capillary column, and then detectors are similar to HPLC, or HPLC detectors are normally used here. Um, so phosphate maybe the buffers like sodium sodium phosphate buffers are used depending on the type of macromolecules you to be migrated. So this is the setup of this is the setup of capillary electrophoresis. So where you can see the two silica tube which is filled with buffer, and it is dipped in the chamber, and both the chambers are connected externally by an platinum electrodes in externally, and it has been connected to the power pack where a high voltage will be applied across the electrode, and it is the buffer solution or electrolyte solution. The ions from this will be migrated into the capillary tube. Depending on the charge, they migrate either to cathode or anode. So as we can, in between the capillary tube there is a detector, so which will be constantly monitoring the flowing molecules here. The solute molecules which are migrating in the silica tube, capillary tube, will be constantly detected here, monitored by the detector, and it will be analyzed. So this is the whole setup of the thing, and the sample will be injected in between the from the inlet of the column, that is in the towards the cathode electrode, towards the cathode electrode. So not the electroendosmotic flow does not disturb here much, as we have seen, it causes the band broadening. So but here the pressure flow of the solvent will in turn, mobility of the ions will also create a hydrodynamic flow, which makes the run faster. Resolution of the component will be faster because of the pumping of the solvent here, pumping of the flow of the pressure driven flow will increase the flow rate, and thereby increases the run of the resolution of the component. So as you can see in this slide, the molecules are transport, when they are coming out, the in between the detector it will be detected, and you can see the eluted whatever the molecules that are detected during the when they pass through the detection chamber. So here if you consider UV visible detector, the constantly illuminating source will be falling on the flow chamber, and the photo cathode tube will collect the transmitted light which is passing out from the flow chamber, and each molecules will be connected. And as you know that the mobility of the each of the individual charged molecule based on their size and charge, smaller the size, greater the charge, migrate faster, as we know, and larger the size and lesser the charge, migrate slow. So, and you can get the plot of absorption if any particular wavelength of absorption seen by the molecule of interest. You can monitor the movement of this elution of these molecules here. So as you can see the time, retention time, and the absorbance on the y-axis. So detectors are normally used for HPLC detectors like UV visible detector and fluorescent. Most commonly used are the UV visible detector and fluorescent, which we have discussed in HPLC system. Laser induced laser beams are used for fluorescent incident light. Okay, so for incident light, the laser beams are used for fluorescent detectors, and even conductivity can be used. But most commonly used are the UV and the fluorescent detectors. And even the mass can be conjugated. Mass detectors can also be frequently used for analyzing the structural information of the resolved peaks. So you can get the even the sequence and the structural information of the resolving molecule here, that can be connected as the capillary tube can be connected to the mass spectrophotometer. So the sensitive detectors are needed as the small concentration of the samples are used. So as you can see here, impurities, you can unknown impurities, if it is there, you will get the peaks. Amino acids like serine, labeled serine, di-labeled, two labeled serine, you can see the shift in the position, the illusion movement of the ions will vary as you see the labeling difference also. And different amino acids you can see, impurities also you can see. So this is how the electrophoresis work. And mainly the advantage of this particular technique is that it is often selective, and it is an alternative to HPLC system. And it is easy and predictable. Selectively you can analyze the molecules, and it has a high separation efficiency, millions of because because the tube is very long, so separation happens very nice, very effectively. Resolution of the molecules happen very effectively, and analysis also happens very precisely, even though a very small quantity of the sample is used. For example, it requires a very small range of sample like 1 to 10 nanoliters, and it gives a faster separation of 1 to within 1 to 45 minutes you can get the results after subjecting for analysis. And it can be made automated, and it can be easily coupled with mass, so that you get more information on the sequencing of the molecule, or structural information can be enabled by coupling with the mass spectrometric analysis. So the disadvantages is that it cannot be made as a preparative scale. So you cannot purify the molecule here. It is only an analytical tool where you can analyze the things, and low concentration of samples, and large volume of buffer will be eluting. So that is a major difficulty. It cannot be extended for preparative purpose. So applications, when it comes, it is oftenly used for nucleic acid sequencing. Normally whenever any DNA recombinant DNA works are done, so if we get any amplification of PCR products, or the sequence of the gene, if you want to analyze, all those sequencing data can be done by nucleic acid sequence can be analyzed. Similarly, the amino acids can be analyzed, not a sequencing, amino acid analysis can be done. And similarly, you can even any charged molecule, for that matter protein, peptides, all those things you can analyze using this particular technique.